So I would like to thank the organizers for inviting us to give this brief presentation. So I'm a biomedical engineer by training, so I've been working on medical imaging for a little bit. So I'm going to start off by telling you some of the use cases why we're doing this kind of work, then I'll leave Jason to tell you how we're getting there. So I'm here, I got invited here to talk about imaging data. So this is a very large database conference, right? So is imaging data really big data? So I'll try to lead you into that and see if we're there yet. So I think most people still recognize this. So this is how radiology used to be done. So you get sheets of film that's printed. So all your case studies would be 1, 2, maybe 3 sheets at most. That's very little data. But we went to the digital age, right? So the age of internet and age of data. So this was a study that was conducted by the Mayo Clinic group in 2014. It was published in 2015. Look at the first decade past the millennium, looking at their data. So they looked at CTs and MRs. You can see there's pretty much an exponential rise in, this is actually mean number of images per exam, and it's just been skyrocketing because in the US, we like technology, right? So we scan as much as we can to get more and more information, even though it's not exactly useful all the time, but we do it. And the other thing is, part of the culture here is a lot of medicine is defensive. If you don't do it, you might get sued and stuff like that. So I just pulled the UCSF data last week. So this is our chart. So you can see, so the blue is CR. CR is computed radiography. So basically that's chest x-rays. So it's digital now. It's not film anymore. Then the next one is CT. Then ultrasound, MR. I put MR PET there. I'll tell you why that is. So as you can see on the top, the number of exams is sort of, chest x-rays is depending on the volume, right? The other ones, if you look at it, is on the rise because we can do a lot more with the computational power that we have. So most of the imaging technologies are highly dependent on computing power, not only by the console, also the post-processing for research purposes. And the other thing to note is the size of the data sets. You can see CT, even though CR is by large number of exams, but CT and MR and ultrasound has a lot more data because those are more dynamic and those have a lot more images per exam. So I'll tell you about a little bit of image types, right? So some of you might recognize what this is. This is the first X-ray image ever taken in 1895 by Wilhelm Röntgen. So then this is a chest X-ray of a kid. Then these are CT images. That's a PET image. So the PET is positron emission tomography. So that's primarily used to look at metastatic tumors. Then I'll show you this. So that's a brain. That's a brain. These are all anatomic images. Then that's a tumor of the brain. That's what we call a diffusion image. That's your arterial, also this is your arteries. Then this is metabolic data. So, but everything here, this is MR. So just one modality can give you, and this is only a fraction of what MR can do. So MR, because it's kind of interesting, it uses spins of protons primarily in doing imaging, so you can actually look at function. So now we're no longer just interested in anatomy. We're interested in function, and function changes when you do interventions. So this is a very powerful tool for sort of personalized medicine, if you will. And where a lot of research is involved with looking at how to better utilize the functional aspects of the imaging. So then those are just the raw images. A lot of the data that we collected are actually processed images. So this is what like the NIH has been funding for the last 6-7 years. This is what we call a connectome. So basically here, this is anatomy. Then this is actually the tractography that we generate using diffusion images. Then what you can do is you can look at the physical tracks and you generate these networks. And from the networks, you can recognize, okay, maybe potentially changed because of diseases, or in this case, is aging. So my active research interest is actually in the developing brain. So we look at very young brains instead of very old brains. Then this is what we call a functional connectome. So basically, this is using the oscillations of your hemodynamic responses. So basically, the oxygen that's carried by your blood, regionally, it actually correlates. So this is the functional connectome. Furthermore, you can utilize the anatomic images to look at your cortical folding, and you look at curvature. You can look at a lot more things. So these are for research. They haven't been integrated to the clinic, but we're trying to figure out how to leverage these data in medical diagnosis. So just brief snapshot. So at UCSF, we have about 6 million, as you can see, exams, about 1.5 billion images, about 1 petabyte, about evenly split between clinical and research. Research is actually slightly more because the process image storage. Then that's just a sort of estimate. But currently I'm primarily in neuro, so in neuro studies, about 800 images for clinical, but we have upwards towards 6,000 for research. Then the largest neuro exam is the PET-MR. It's 72,000 images on average. This is because we collected over a longer duration of time. So why I left the PET-MR here is interesting is because you saw the chart that was early on. PET-MR is by far the smallest number of exams, but you can actually see it on data size. So compared to CR, which is chest x-rays, they're at about 1/50th of the number, but they're only at a third of the total data storage. So they actually acquire a lot more data storage that needs to be processed and stored and curated, right? So it's very interesting. We haven't really got into how to leverage the PET-MR data because it just came online literally about 3 years ago. So I'm going to tell you a little bit of use case before turning to Jason on how we actually going to try to get all the data together. So I'm going to show you 3 cases. So this case is actually, someone complained to Somali about acronyms. So I'm going to start with telling you what CMB is cerebral microbleeds. So my colleague Janine Lupo, so she has a group doing tumor research. So this is looking at radiation treatment. So radiation treatment induces cerebral microbleeds in the long term. So this is looking at, okay, where do those microbleeds occur and how do they affect your brain function? So next, this is my colleague Peter Larson's group. So they've been looking at, so this is actually generating CT-like images from MR because traditionally PET is acquired with CT, but CT has very poor soft tissue contrast, but it's great for correcting for bone. So his group has been trying to use deep learning methods to sort of compute the CT-like images to correct for those bone attenuations for PET studies. And on the right here is synthetic MR. You can actually generate sort of contrast that's similar to what we are used to, T1, T2s. Those are image types that the radiologists are used to by acquiring less data. So one last case is, this is my colleague Shamala Majumdar's group. So they've been working on looking at basically automatically classify knees and bones and cartilages. So this is try to figure out, okay, how do you grade osteoarthritis and predict progression of disease? So in order for the projects to happen, you sort of need to sort of collect the data and so more data is always better, right? And so we need to sort of figure out a way to leverage all the data. So I'm going to leave Jason to tell you about how we're trying to do it at UCSF.
So I'm going to carry on here and describe what we're doing institutionally at UCSF to build up what we're calling the imaging commons to bring our medical imaging record into what we're calling a broader information commons, where it can be joined in a shared research and data discovery platform and joined with clinical data, notes, EMR data, and so on. This is our team that's working on this. It's sponsored by the Institute for Computational Health Science, Department of Radiology, and the Center for Digital Health Innovation. Just want to point out, Sharat and Eugenia Rutenberg from ICHS. Rick Larson is the Director of Research Informatics at UCSF. Ed and Joe are technology directors at CDHI, and then we've got our data science and computer science team, consisting of James, James Beck, Boris, who's in the back, and Ritika. Okay, so as Duan pointed out, there's a lot of computational research that's promising to advance image classification and treatment outcomes, but it requires access to multi-domain data and a lot of computational resources and data science tools. And so as the previous speaker pointed out, scientists are spending a lot of time accessing the data, classifying it, just in order to conduct their medical research. And so what we're trying to do is build up this information commons to solve these problems once for everybody. Put all the data in one place, imaging, clinical, so on, make sure it's HIPAA compliant, de-identified, and then searchable, and then layer on all the computational resources that would be necessary to do scientific studies together with the data science tools. And the imaging commons is one module, specifically to integrate the de-identified imaging data into this broader information commons pool. So just as a motivating scenario, just an example of how researchers are often conducting their research today. And so this example is a research group that's trying to correlate the extent of surgical resection for patients with brain tumors to the patient's outcomes. So the first step is the research group has to find their cohorts. So they get an IRB, they look up the pathology notes, and they have a research associate reading through those notes, finding patients with the appropriate diagnosis, and then they have to go back and find the images. Those images are in a different location. They have to extract those from the clinical packs to obtain their volumetric parameters, and then they ultimately want to correlate those with additional parameters in the EMR, and so somebody has to go back to the EMR and again manually extract those, look up all the information. So overall it's just a very labor-intensive process to access the data, find the study parameters, and so on, even for a moderate study. So it's a painful process to, one, access the data. To identify data in the hospital is available only through, it's restricted access, so you have to go through either an IRB approval to gain access to it. And then the other thing is data from different parts of the hospital are in different data silos, so you have to go through separate mechanisms to gain access to that information. And then once you have the data, it's often a very manual process for a lot of researchers to extract the information of interest. And this process is just tedious and is really slowing down the research program. So we're trying to bring all the data, again, as I said, into one place, de-identify it, and provide the computational infrastructure and data science infrastructure, solve this problem once for everybody. And less time data wrangling is more time doing science. So this is sort of a modular picture of the information commons and how we're building it up. Very high level picture. We have some shared storage, and domain by domain, we're bringing the data into the information commons. So clinical notes, bring in, we go through a de-identification pipeline specific to the notes. We look replacement identifiers up in an ID mapping service. We find patient-specific date shifts. we de-identify the data and store the data in our shared storage, and then extract parameters and concepts from those notes that we promote to a searchable data layer. And Gudolf, who's in the back, is involved with this in extracting UMLS concepts, for example, from notes. Structured EMR data, similar process, de-identify it, and then the imaging data has its own de-identification pipeline. We extract searchable attributes from the images after the de-identified images have been stored, and we promote those into a search layer. And then we're just repeating this for different data sources at the university: genetics, sensor data, and then annotations and labels are of course very important. And then once we have all the data in this common pool and searchable, then we're layering on top of that a data discovery layer with NLP tools, machine learning tools, and so on. A little more detail on the imaging pipeline. So we're getting data from different data sources. This can be scanners, clinical scanners, research archives, or clinical packs, and so on. These generate Dicom images, Dicom is the standard data format for medical images in the industry. These come in, go through a de-identification service. We store all the original attributes in case we need to re-identify the data and return it to the clinical workflow. But we look up replacement identifiers, de-identify the data, archive it, store the source data in the shared archive, extract our attributes and promote those to the discovery layer where they can be merged with attributes from other data domains. Okay, so the challenge here is that HIPAA and other privacy regulations restrict how we make that data available to the research community. Clinical data can't be used for research without either an IRB approval or unless it's been de-identified. HIPAA does define what needs to be removed from the data. PHI, specific identifiers like dates, MRNs, addresses, I think everybody's familiar with these, need to be removed from the information, from the health information for it to be considered de-identified. And it's really important for us to get this classification right because it impacts who can access the data, how it's accessed. For example, do you need an IRB approval to gain access to the data? And then also architectural decisions like where and how the data is hosted and what kind of security controls we have on that data. The problem is that a lot of the clinical data is unstructured or only semi-structured, and so it's really hard for us to know with 100% certainty if a data set that's gone through a data identification pipeline is actually 100% de-identified. And so we have to go through what HIPAA calls an expert determination analysis to determine if the re-identification risk of so-called de-identified data is sufficiently small that we can classify it as de-identified. This slide just shows the structure of the medical imaging Dicom data set. So the imaging data consists of a header with meta information and acquisition parameters, and then the pixel data. The header fields are defined by the Dicom standard, and they're identified by group numbers and an element number. And the standard tags are ones with even group numbers, and ones that I've shown as examples in red are fields that we know have PHI in them. Other fields may or may not have PHI, we don't really know up front. And then there are private tags that are used by the vendors. We also don't know what's in those. And then we also run across rogue tags, which are neither in the standard or odd groups. And again, we don't know what's in those. So all these have to be looked at. And then in addition, there's the pixel data, which has the imaging information. But in many cases, you'll find additional text information burned into the images. And this may or may not include PHI. So we have to build up this de-identification pipeline. Dicom provides some guidance on how to do this. So we basically start with our identified data set, look up synthetic identifiers to replace the patient name and IDs and so on. We store the original attributes for later re-identification. And then we replace the PHI with our synthetic identifiers. We remove all of the private tags except for a set of whitelisted ones which we know are essential for analysis but don't contain PHI. But after that, we still need to check for PHI in non-standard locations, and then also consider the possibility that PHI exists in varying forms. So there are going to be name variations, there are going to be dates of service scattered throughout the data set, and so we need to perform fuzzy matches to detect all of those and replace them. And then finally, you have the PHI in the data, and that either needs to be masked or OCR to determine if the text in the data is actually PHI. And so we can mask that out. So some examples of imaging data with burned-in text. So ultrasounds tend to have text burned into standard locations, which is helpful because we can identify that and just mask those regions out uniformly. However, all the images still need to be interrogated to see if there's a PHI in non-standard locations. And so we've developed an OCR pipeline. It involves some image pre-processing. We start with the input image, do some edge detection, some morphological operations. We threshold those regions out, try to identify the bounding boxes where there's potential text, some additional filtering and masking on those regions, and then we OCR the data to extract the text regions, and then run it through our PHI detector. If it has PHI in the text, then we quarantine the images rather than masking out the regions. And if it doesn't, we pass the data on to our archive. So far, we've validated this pipeline with 1,000 manually reviewed exams, and we've achieved 100% accuracy in the detection of PHI in the pixels, which is good. We're getting a lot of pre-processing positives, which slows our pipeline down but doesn't affect our accuracy. And we're in the process of undergoing an expert determination analysis on our full pipeline using 60,000 randomly sampled exams from our clinical packs. And we're projecting right now that it's going to take us between 30 and 60 CPU years to identify the entire clinical imaging record of data that was acquired since 2000. So, it's doable. And so once we have the data in the info commons, then we can start to build up tools. So we can build up very simple user-friendly tools. An example shown here is our image query interface, and we can ask simple questions like, show me all the data from post-operative exams from for brain MRIs. And click search, and we can get that list of de-identified data sets, and the users can visualize the data. We can also then directly query any of the searchable imaging attributes, and then ask questions like, show the display, the distribution of imaging modalities for each of the body parts that have been imaged. But then, the more interesting questions involve joining the imaging data with data from other aspects of the patient's record in our information commons. And now we can start to ask really interesting questions like, give me all the CT images for patients where there's some reference to squamous cell carcinoma in their notes, and they have a diagnosis code, a particular diagnosis code. And so you can formulate that pretty easily, ask the question, and then within a matter of seconds to minutes, we can get a list of images that fit that profile, and then we can go on to apply our machine learning algorithms or other data science tools to those data sets. And now we've shrunk this pipeline from from months down to minutes for our researchers. So just in summary, we're integrating all of the entire clinical record, notes, EMR data, imaging data into a compliant shared data pool and discovery platform to support shared health science research. We're going to bring in data from other domains in a modular fashion. We're spending a significant amount of effort to prep the data to go into this platform, but we feel like it's a worthwhile effort because it's going to save a lot of researchers a lot of time and stimulate the ability to ask questions very easily of our data set. And then I just wanted to thank all the people that have been involved in this project, the Imaging Commons Group, CDHI, Information Commons Steering, Department of Radiology, and then we have user groups that are providing use cases and scientific guidance as well. And thanks for your attention.
Did I misinterpret that the de-identified data is available without IRB?
That is the plan, yes.
Okay.
Yes. Once we get expert determination complete, and we're certain that UCSF Privacy Office can classify the data as de-identified, then it's not restricted. However, we will still impose access controls on it, and those are being developed.
I see. So what about the notes, the EMR notes?
It's a similar situation. There's a de-identification pipeline that's been developed and is being validated statistically, also with expert determination analysis. And that's very close to being completed. So, yeah.
Hopefully nobody figures out how to compute a face from a brain.
Exactly. So in some cases, yeah, we do have to. It's not required by HIPAA right now, but there are methods, for example, to strip the skull off of the images so that the facial reconstruction can't be done. But if we have to do that, that's going to add a lot of complexity to the pipeline.
So one question, you did mention a little bit about generating some kind of data from your images, but I was wondering if there's some way to summarize them, to measure volumes consistently across them, or otherwise compute summary statistics that would allow you to not have to store all the pixels in order to actually do some kind of a study. Is that possible? Or everything goes back to the pixels anyway?
I think we certainly want to store annotations and derived quantities that have been extracted from the images, and those would be annotations or labels. But I think we still want to have the source data available in the information commons, because people are always going to want to do new things with the source data. But those quantities, once they've been calculated, they can be valuable to other researchers.
And along the same lines, do you have a need to, or is there some way that you, within Dicom or some other way that you ensure the integrity of the images? Is there a checksum? Do you sign them in some way and track the provenance as they're going through this process?
Through the de-identification pipeline, for example?
Yeah, and then eventually through the research.
Yeah, I mean, we know that we're getting the data intact from PACS just through the Dicom protocol. We have some assurance that we're getting the original data intact. Yeah, data provenance is definitely an issue for derived data that's going to go back into the information commons. I think we're pretty confident that the source data is intact as it comes from PACS. If there are questions about the header or information that's been removed, we can always go back to the original data because we store all the original attributes. But for derived terms, the provenance is going to be very important, so people have, one, the ability to understand what the pipeline was, how those quantities were derived, but also so people have some confidence in assessing the quality of results. Who generated them, what software was used to generate those derived results, and so on.
So in one of the slides, there is a OCR Tesseract.
Tesseract, yeah.
Yeah, so that's the only Google product, or is everything else is also done on Google?
That's the only Google product in that pipeline, yeah.
And for DID, you're not using like DLP or anything?
We're not using what?
DLP, data loss prevention?
No.
Okay.
No. Actually, I'm not familiar with it. What is?
It is a Google acronym for a service which they offer for DID. Yeah.
Okay.